So good afternoon, everyone. Hugo Fernandes from the University of Coimbra, and I'm here to present the GILDED project, a novel therapy for the treatment of diabetes. Recent data on diabetes has been coming out, and as of 2025, I have to say that looking at the data, I was quite surprised to know that half of the people, or close to half of the people living with diabetes, they do not know that they have this condition. So the diagnostic is very poor. Diabetes is mostly a disease, not only, but mostly a disease of beta cells. So beta cell dysfunction is the main driver of diabetes. Beta cells are the ones responsible for secreting insulin in order to control the levels of glucose in the blood. Most of the current therapies in the market, the multi-billion-dollar market, are actually targeting the symptoms and not really the root cause of the disease, which is the beta cell dysfunction. So enter our project. We have developed in the lab a screening assay, a screening platform that allows us to mimic conditions usually found in diabetic people, that is high levels of glucose and free fatty acids, namely, for example, in obese people. So we have screened a library of microRNAs in this case. We can actually have screened something else, small molecules. And we actually found that when you expose these cells to the conditions that we mimic in the lab, these cells die within 24 hours. So only 25% of the cells survive. However, if you transfect the cells with some of these microRNAs, not only you actually rescue the cells, but you can actually have these cells proliferating despite the fact that they are exposed to high levels of glucose and fatty acids. And the effect is huge. Those of you that have done high-throughput screening, they know that probably we are looking at 3 times standard deviations for lead candidates, whereas here we have actually 3 times the mean. So these cells are really proliferating in the plates. We went on to test in patients the expression levels of the microRNAs that we select as the best microRNAs in our screening. So there is a condition where people are exposed to high levels of free fatty acids and sometimes glucose, which is people undergoing bariatric surgery. So people selected for bariatric surgery, we analyzed their expression of these microRNAs, and actually we found that those obese people that are not diabetic, they have higher levels of the microRNAs that we identified in our screening, suggesting indeed that they can confer some sort of protection, even though that these people are morbidly obese. So there's one particular feature. So we selected one of the hit microRNAs for further studies. We characterize this microRNA. We know the mechanism of action. But there's one particular aspect that I would like to draw your attention to. This microRNA upregulates GLP-1 receptor. It is the same receptor that is used by these multi-billion-dollar drugs called the obesity drugs in the moment. So our microRNA can actually upregulate the expression of the receptor that is used by these molecules. If you don't have the receptor, you also don't have the effect. So having this great microRNA is not the full story, as you probably know. We actually need a delivery system. So we are currently working on a lipid nanoparticle formulation that encapsulates this microRNA. And we are further functionalizing this lipid nanoparticle with an antenna that targets the lipid nanoparticle to the GLP-1 receptor, which is expressed in the beta cells. So in summary, what we have now is a microRNA that we believe is quite game-changing in the treatment of diabetes. We are developing a formulation that is able to deliver the microRNA into the pancreas. And there's one important aspect that I would like to draw your attention. Because the microRNA upregulates the same receptor that we are using for targeting, we can actually envision a situation where during treatment, we can actually start to reduce the dose while maintaining the efficacy of the treatment. And therefore, this is a self-amplifying mechanism where the burden and the cost for the patient can be decreased. So we actually need the help of the George Lewis Award and the Tecnimede in order to further develop, especially the nanoformulation system with the antenna, to deliver the microRNA into the pancreas. And therefore, that's what we would like to do in the next 6 to 8 months. And later on, we also want to test it in vivo models of diabetes, which are already running in the lab. So we have a team of people that is able to analyze the cellular and molecular parts, the drug delivery systems, and to keep us in check, it's always good to have scientists come to the earth, and we have a clinical endocrinologist that keep an eye on us. So we hope to make diabetes the first metabolic disease that is not just managed, but can actually be reversed. So thank you very much for your attention.